Okay, so the next process that we're going to talk about is known as translation. In translation, the RNA molecule that we just made during transcription is now called a messenger RNA molecule. This messenger RNA molecule basically contains the code sequence for how to produce a protein. So in translation, the messenger RNA will be translated into a sequence of amino acids, and this sequence of amino acids is what will eventually dictate the formation of a protein. So one of the most important components of translation is the ribosome. The ribosome is the site of protein synthesis, so obviously all parts of the translation process are going to occur on the ribosome. Now, between eukaryotic and prokaryotic cells, the ribosomes do look quite different. Generally, the rule is that prokaryotic ribosomes are much smaller than eukaryotic ribosomes. Eukaryotic ribosomes are basically composed up of two small subunits as well as prokaryotic ribosomes. In prokaryotic ribosomes, the large subunit is 50S in size, and the small subunit is 30S in size. These subunits come together to make a 70S ribosome. In eukaryotic ribosomes, the large subunit is 60S in size, and the small subunit is 40S in size, therefore coming together to make an 80S ribosome. So after transcription has ended, the messenger RNA molecule that we see here, the single-stranded messenger RNA molecule, is going to bind with a ribosome to start protein synthesis. So basically what's going to happen is this ribosome here is going to read this sequence and cause the formation of a protein after that. So the way that the ribosome works is it reads the messenger RNA molecule in three-letter sequences called codons. So right here we have a three-letter sequence, that's a codon. Here's another codon, and another codon, and another codon. Before we were kind of talking in the language of letters, just single nucleotides, but now we're actually grouping them into three-letter sequences. So now we're kind of talking about words essentially. So basically the ribosome is going to read the words, and then it's going to allow for the proper incorporation of an amino acid. So how do these codons actually correspond to amino acids? There's actually 64 codons that get broken out between the 20 amino acids. So between the 20 amino acids, they can all have a variety of different codons. So a simple way to look into this is by using a chart such as the one depicted here. We have all the different codon sequences listed and the corresponding amino acids that they represent. So before we talk about how we actually produce this amino acid chain, we have to talk about where the amino acids actually come from. The amino acids are always held in the cytoplasm, bound to an RNA molecule called transfer RNA, which we see here. It gets its name ironically because it does look like a T. So basically this transfer RNA has a very important function. Its function is to bring the amino acids from the cytoplasm to the ribosome so that they can be incorporated into the growing protein chain. So there's a variety of different transfer RNAs in the cytoplasm, and they are all charged with one of the 20 different amino acids. This one's charged with alanine, but there's all different types of amino acids that they can be charged with. So on one end you have it charged with an amino acid. On the other end you have something called an anticodon. So the anticodon is basically the complementary base pair to the codon that's found in the messenger RNA. So with this one being alanine, its anticodon is CGG. That means it's going to directly correspond with the codon in the messenger RNA that reads GCC. And this will be really important as we talk about in a second, and kind of a proofreading and double checking mechanism as we actually go through the process of linking the amino acids and building the protein chain. Okay, so as the ribosome is reading through the three-letter sequences that we now know are called codons, it's going to come across a sequence that says AUG. Now AUG always represents a start codon. A start codon is where we actually are going to start translation. So any codons before AUG are not going to be incorporated into the final product of the protein. We only start translation here at this start codon. So if you look at the chart that we looked at earlier, we can see that AUG codes for an amino acid known as methionine. So as the ribosome reads this AUG sequence, we know that that codes for methionine. So the next step that needs to happen is a transfer RNA molecule that is charged, in our example now with methionine, is going to come over and it's going to deposit its amino acid here at the ribosome. Now in order to double check that that is the appropriate place for that amino acid to go, it does need to bind its anticodon sequence to the codon sequence here. So if the codon sequence reads AUG, our anticodon sequence is going to read UAC. If those match up appropriately, again, methionine is going to be deposited here. Then what is going to happen is this ribosomal structure is going to shift its frame to the next three letter sequence, which is again our next codon, and it's going to read UUU. So again, what you have to do is go to your chart and see which codon sequence codes for that specific amino acid. So if we look for UUU, we find it here. UUU is a codon sequence that codes for phenylalanine. So again, what is going to happen is this transfer RNA that's coded for this example with phenylalanine is going to come out of the cytoplasm and up to the ribosome. It's going to bind its anticodon sequence to the codon sequence, which since here the codon sequence reads UUU, the anticodon sequence is going to read AAA. Again, if everything matches up, the phenylalanine is going to be deposited here at the site of the ribosome. What's going to happen is the phenylalanine is going to form a bond with methionine, and this is known as a peptide bond. So what we're going to basically do is start peptide bonding these amino acids together until we finally get our entire protein. So again, this ribosome is just going to keep shifting down the messenger RNA transcript in these three-letter codon sequences. It's going to read the three-letter codon sequences, and it's going to make sure the correct transfer RNA comes and incorporates the appropriate amino acid into the growing protein chain. Now, this doesn't go on forever. This will stop when we get to something known as a stop codon, which we see here. Okay, so there are three stop codons, and if we look at our chart again, we can see what the stop codons are. The stop codons are UAA, UAG, and UGA. So basically what happens, anytime the ribosome comes in contact with a stop codon, translation will stop. The thing is that with the stop codons, there's no amino acid that corresponds to the stop codon, so we don't have any amino acid that's going to be incorporated there. So when the ribosome comes along to a stop codon, it's basically going to dissociate and fall off the messenger RNA transcript. But at this point, we have already produced our final amino acid linkage here that leads to a protein. So for our example, this messenger RNA transcript dictated the production of a protein that read methionine, phenylalanine, leucine, alanine, tryptophan, serine, and proline. And then again, this was stopped here. Now, this isn't the complete end to the production of a protein. There are several kind of secondary and tertiary folding mechanisms that does need to happen. But this does show in translation how we go from messenger RNA into an end product, which is a series of bound amino acids that forms the protein.